Fatal familial insomnia is a rare inherited prion disease. Clinical insomnia typically involves a failure of sleep initiation, where the patient's biological capacity to sleep remains intact. FFI is a distinct pathology, a progressive neurodegenerative obliteration of the brain's internal sleep-generating mechanisms. The disease physically destroys the brain's sleep-wake architecture, dismantling the neural pathways until the act of sleeping becomes biologically impossible. This trajectory is absolute. It begins with a single genetic mutation and terminates in total thalamic failure, complete autonomic collapse, and inevitable death. The sleep loss in FFI represents the outward sign of a brain whose anatomical hardware is being systematically dismantled. This sequence shows a point mutation in the PRNP gene. Inheriting a single copy initiates the disease. During transcription, the protein chain folding incorrectly into a knotted structure. These prions trigger a chain reaction, forcing healthy proteins to aggregate into dense clusters throughout the brain. As these prions accumulate, they cause localized, progressive neuronal cell death. The brain tissue begins to degrade at a microscopic level. In sporadic fatal insomnia, patients develop the same symptoms without a family history. This indicates that the prion mechanism itself is the primary driver of the pathology, regardless of whether the mutation was inherited or occurred spontaneously. A microscopic protein folding error acts as the catalyst for macroscopic brain failure. This is the thalamus, the brain's central hub for sensory relay and the coordination of consciousness. The prions in FFI selectively target this region. They accumulate in and destroy the thalamic nuclei. Under normal conditions, the thalamus generates rhythms to transition the brain to sleep. As neurons die, glial cells proliferate, creating dense scar tissue known as gliosis. This erodes structural integrity, causing communication between the thalamus and cortex to stutter and vanish. This gliosis physically dismantles the anatomical nuclei required for sleep generation. The pathways necessary to regulate the sleep-wake cycle are effectively erased. The brain cannot execute the biological function of sleep because the neurological architecture required to support it has been removed. This chart shows normal sleep architecture, defined by distinct markers like sleep spindles and rhythmic slow waves. The FFI data on the right shows initial fragmentation replaced by disorganized microsleep episodes. Eventually, all diagnostic markers vanish, and the recording completely flatlines into unstructured, low-amplitude noise. This diagnostic erasure dictates the limitations of medical intervention. Standard sedatives and sleep medications are ineffective for FFI patients. They cannot restore sleep. Pharmacological sedation relies on the existence of intact neural circuits and receptor sites. Without the underlying thalamic architecture, these chemical interventions have no physiological target. Stage 1 of FFI is defined by progressive insomnia, alongside the onset of anxiety and panic attacks. Through stages 2 and 3, sleep disruption becomes near total. Physiological markers for heart rate and blood pressure spike as the patient experiences hallucinations. This occurs because the thalamus also regulates the autonomic nervous system. Its destruction triggers a systemic failure. In stage 4, sleep is impossible. Autonomic instability peaks, manifesting as uncontrollable tachycardia, hypertension, and hyperthermia. Death predictably follows, typically occurring between 6 and 36 months after the first symptoms appear. The ultimate cause of death is the complete collapse of central autonomic regulation, resulting from structural brain failure. The causal chain is direct. A genetic mutation leads to prion accumulation, causing thalamic gliosis, the loss of sleep architecture, and ultimately autonomic failure. Sleep is a critical biological function required for survival. Fatal familial insomnia provides the clinical proof of the thalamus's role as the essential regulator of human homeostasis. Without the structural capacity for sleep, the brain loses the ability to coordinate the autonomic nervous system. Sleep is an active, structurally dependent imperative. When the brain loses the ability to sleep, the body loses the ability to live.